Hello, I'm Noel McCarthy, and I'd like to start by saying how pleased I am to participate in this seminar looking at campylobacter research enabled by the PubMLST database infrastructure over the past 2 decades, and in particular the memorial that it is to the work of Ali Cody, who started in the Maiden Group in 2004 when I also joined the endeavour as a PhD student, and having worked alongside each other for the following 16 years. Over the past 2 decades, campylobacter genome sequencing has started and exploded, with over 52,000 isolates now accessible on PubMLST with whole genome data. The huge benefits from these increasingly easily obtained data and their analysis are, however, accompanied by gaps in robust sampling approaches to generate them and the collection of other dimensions of data. Indeed, it can be quite genomocentric, with all other data sometimes being termed as mere metadata to accompany the genome. To my view, this extreme focus on the genome, with relegation of other aspects of data to mere metadata, is ill-conceived. We would be better to focus increasingly on the broader aspects and dimensions of data, no doubt including genomic data, but also, for example, accurate recording and sampling based on time, place, and person, considering different countries, considering domestic versus travel associated, urban versus rural, amongst the place characteristics, and for personal characteristics, the pattern of symptoms patients have suffered, the exposure histories that they can offer. As evident on the Eventbrite invitation to this seminar, PubMLST very much considers these wider dimensions and had a picture not of a genome, but of the geographical distribution of isolates on the database. I'd like to illustrate these arguments with 4 examples, starting with this genome-wide association study of campylobacter genetic markers with patient-reported symptoms. Using a standard analysis pipeline on over 2,000 genomes, software limitations meant running the analysis in batches of 200 to 500 isolates at a time. Looking at results across these batches showed recurrent hits in one very narrow area of the genome, so the genome-wide association study picked that out. But associated with what? The phenotype is again quite complicated, looking across the range of symptoms reported by people, and this association is with some, but by no means all, of these symptoms, emphasizing the importance of a well-characterized detailed phenotype, equally to a well-characterized genotype. Using an approach developed and validated by colleagues in Public Health England, we were also able to look at antibiotic resistance in this same Sentinel surveillance scheme dataset, this being a particular interest of Ali Cody's. Here we see high levels of resistance against, for example, fluoroquinolones and tetracyclines, particularly among isolates from chicken. This became particularly important when the funder decided they wanted to fund more work on antibiotic resistance and proposed cutting our project. We were thankfully able to advise them that with this approach, we could provide lots of information about antibiotic resistance, and they instead increased our funding. This allowed us to expand the extent of calf and lamb liver sampling, as shown here, giving robust estimates of resistance levels there. The additional funding also allowed us to look back in time using archived isolates from 1997-8 and 2007 amongst humans, and therefore we could look at the changes over time as well as the distribution across different animal species, showing, for example, the dramatic rise in fluoroquinolone resistance from levels as low as 5.2% in 1997-1998. Again, the overall point here is the value of having these very different dimensions of sampling and of data analysis. My next example is a simple analysis, but with the advantage of 3 dimensions of data: the place, Australia or Europe typically, the person, meaning which host species the campylobacter populations came out of, and the genome of campylobacter, saying were these populations similar to each other or quite different. Starting at the extreme right, the population coming from sharp-tailed sandpipers in Australia was very close to that coming from dunlins in Sweden. Similar species of bird with a similar shore-feeding wading lifestyle. Again, to the bottom, starlings in the UK and Sweden are closer to each other than to anything else in terms of their Campylobacter population genetics. And up at the top left, the agricultural niche, as mentioned by Sam, showing through with isolates in a chicken in the UK rather more similar to a cow from a UK farm than any of the other bird species. Overall, a simple analysis, but with the advantages of 3 distinct dimensions to help us to learn quite a lot. My last example is going to look at the source of human infection as determined by population genetic analysis, and will build from this systematic review that Fran mentioned, where Ali gathered all the studies that had used those approaches and published in the literature. And I want to focus on the fact that when we looked at these studies, they were all biased. Several of them had actually measured whether their algorithm was a fair algorithm or had a bias in it. And when that was done, they were always somewhat biased, sometimes very. But none of them had then properly adjusted their final results to take that bias into account, including our own work. So with that starting point, we said, well, let's try doing that better in this more recent study. And with Melissa van Rensburg, worked out a way to adjust for the bias. And we found that that substantially increased the proportion that we attributed to chicken for Campylobacter jejuni, but not for Campylobacter coli. Having done that as one way, we then said, well, are there other quite independent ways we could answer the same question? The complementary approach that we used was an analysis restricted to those isolates where we were very certain of their origin. We were certain because when we consider all the isolates that come out of, for example, a chicken, for about 60% of those isolates, well, we find the same similar isolates in wild birds or particularly in other farm animals, echoing Sam's talk. But there is approximately 40% of them, which we really only find in chickens and in humans. So we're quite certain when we see one of those types that that originated in a chicken. And indeed, that proportion causes this much of the human disease. And similarly, we can get about 30% of cattle and sheep types are very typical of cattle and sheep, and they cause a rather smaller amount of human disease. So starting with that, we can then think of extrapolating. So having observed that this proportion of chicken isolates causes this part of human disease, where we actually know that they're very much chicken types, we're then going to assume that the 60% here are equally likely to cause human disease as these 40%. And that gives us this portion up here, which combined suggests to us that about 75% of human disease is originating in chicken, which very much echoes the results we got on the properly adjusted population genetic analysis. Similarly, if we're saying that the 30% of ruminant isolates causes this much disease, an extrapolation to consider what these might be doing would suggest that the total amount coming from ruminants is up here. And let's step back from that overall extrapolation to these specific isolates where we're quite confident of knowing their origin, be that be from a chicken or from a cow or sheep. And we'll consider these origins inferred from the testing that we did in the lab using the same colors as before. For example, those with a genetic code suggesting they were from a ruminant source in brown. And we'll additionally consider what those people reported as having eaten in the week before onset. And here we're looking at whether they reported eating calf liver, lamb's liver, or chicken. And we can see quite marked differences. Very few reported eating calf liver. A lot of people reported eating chicken, more so if they were infected with a chicken type or an unknown type. But perhaps most striking here in the middle, of those who had a ruminant type, fully 23% remembered eating lamb's liver in the past week. And the odds of this memory was 15 times higher in those with this type compared to those with the chicken type. A strong association between our later lab estimate of the origin and the human memory of what they'd eaten. If we extrapolate from this to total numbers of cases, we can estimate that between 5,000 and 10,000 human cases have an origin from lamb's liver. But for this example, the real focus is on the power of joining, in this case, genetic and exposure history analysis, as well as a sampling frame that includes human and well-sampled animal populations to let us draw inferences. So my conclusion is again the importance of using the range of different dimensions of data when analysing and indeed planning sampling, and not getting overly focused on genetic data to the neglect of other aspects. PubMLSD is well set up to include both these genomic data with all the other dimensions subject to people submitting those data and letting that be accessible to a wide group of people to jointly analyse. So with that, I'll finish, and thank you again for your attention, and I suppose particularly again to remember Ali and her contribution over these 2 decades.